Okay, thank you. Good morning. So the study I want to present to you now is actually in situ isotopic analysis of carbon isotope in organic matter from the cherts of the Dresser Formation in the Pilbara Craton, which are dated to 3.5 billion years and contain some of the oldest evidence for life on Earth. So the Pilbara Craton is well known not just as the site of current stromatolite, but as the location of Earth's oldest putative stromatolites and microfossils. The beautiful stromatolites of the Sturley Pool Formation, dated to 3.4 billion years. These are probably of biogenic origin. I don't think a lot of people dispute that now. Slightly older are the putative microfossils that were found in the famous Apex chert, whose biogenic origin has been debated for over 30 years now. The oldest putative stromatolites and microfossils occur in the bedded cherts of the Dresser Formation, dated to 3.5 billion years. Although the biogenic origin of these oldest structures have also been debated, there are isotopic evidence that support microbial sulfur disproportionation and microbial methanogenesis in these rocks. And another important observation is the occurrence of organic matter in association with these alleged biogenic structures, which also support some kind of link that these are actually produced by biologic activity. Bulk carbon isotopic analysis of the organic matter that is preserved in these rocks have shown that it is depleted in carbon-13 in a manner very similar to that of organic matter produced by autotrophic microorganisms, suggesting that it is of biogenic origin. However, experimental studies have shown that similar isotopic compositions can be obtained during abiotic carbon fixation in processes which are similar to the industrial Fischer-Tropsch type synthesis, which I will talk about a little later. And therefore, they concluded that carbon isotopic composition of organic matter by itself is not a conclusive evidence for a biogenic origin. In an attempt to better constrain the origin of the organic matter preserved in these rocks, in these cherts, we have decided to apply in situ carbon isotopic analysis of the organic matter. And this technique has the potential of distinguishing between different generations of organic matter in the same sample, like you see here from the Sturley Pool Formation, a work by Lepot et al. They have found within one sample organic matter in the form of these globular clusters and spheres, and they have shown that it has slightly different carbon isotopic composition. Now, if you would do bulk analysis for this sample, so you might just get an average, which kind of obscure the real picture. So like the work shown here, our work was also done in the Wisk-Sims lab, which you have probably visited on Sunday. Now, we have used a standard of natural chert that contain organic matter, which allow us to accurately correct the instrumental mass bias. Unlike previous works who have targeted microstructures in this sample, which have used crystalline graphite, which now is known to have a different instrumental bias. So our samples come from the Pilbara drilling project, Drill Core 2, which have penetrated the Dresser bedded cherts at depths of about 80 to 90 meters below a section of pillow basalts, providing relatively fresh samples to work with. Our samples come mostly from the bedded cherts at the upper part of the core, but also from a hydrothermal chert vein at the bottom part of the core. And so samples were cut into thin sections and studied under reflected and transmitted light before the analysis. So here you see thin section of bedded chert from the uppermost part of the section, which yielded the most diverse assemblage of organic microstructures. And we have identified 5 main types of organic microstructures in the samples. Layers of dispersed organic matter, amorphous organic matter clots that occur in these rich, organic matter rich beds, and these were designated type 1 clots. And there was another type of clots which had a more distinct shape and occur mostly in the organic matter poor beds, and these were designated type 2 clots. And except for those, we have also found organic matter which was concentrated along stylolytic fronts and organic matter that is preserved in the core of carbonates. So here are the results of our carbon in situ isotopic analysis. Over on the right, sorry, you see images of individual analysis pits in selected microstructures and the isotopic composition that was obtained for these specific pits. And overall, that you see that the overall range of isotopic composition in this sample is over 13 per mil. Over on the left, you see histograms which present the distribution of isotopic composition in specific organic microstructures. And the most interesting observation here is the bimodal distribution of isotopic composition in the clots between the type 1 clot, which had an average isotopic composition of minus 28 per mil, and the type 2 clots, which have average isotopic composition of minus 34 per mil. Here you see individual analysis isotopic composition, delta 13C values, plotted against the 13CH over 13C ratio, which was also measured during the analysis. And this ratio served as a proxy for the sample's hydrogen-carbon ratio. So there is no correlation seen between these two parameters, which is important for 2 reasons. First of all, it indicates that the range of isotopic composition that we see is not the result of a variable instrument bias that might arise from varying hydrogen content in the samples. And second, during thermal maturation of organic matter, it can change its isotopic composition by preferential loss of isotopically light methane. And this process usually leads to a negative correlation between isotopic composition and hydrogen-carbon ratio, which is not seen in the samples. And therefore, we interpret that the range of isotopic composition seen in the sample is an original heterogeneity and not the result of later thermal alteration. Here is the sample from a hydrothermal chert vein. And in this sample, the only organic microstructure that we have identified is these organic matter clots, similar in appearance to the type 1 clots, which I have shown in the bedded chert sample. And isotopic composition in these clots are also similar to the isotopic composition of the type 1 clots in the bedded sample, with an average of about minus 28 per mil. So these textural and isotopic similarities suggest that the clots, both in the bedded chert and in the hydrothermal chert vein, have the same origin. So what is the origin of this organic matter? So as I said before, some works have suggested that the organic matter could have formed by abiotic processes similar to the industrial Fischer-Tropsch type synthesis. In this process, inorganic carbon, mostly in the form of carbon monoxide or carbon dioxide, is reduced by hydrogen over the surface of a metal catalyst. And this process can form simple chains of hydrocarbon. However, the correlation that I've seen between isotopic composition and specific microtextural types is hard to reconcile with this process. If the source of organic matter was just simple polymerization of hydrocarbons formed this way, we would have expected it to have a uniform isotopic composition, at least in one single sample, which is not the case. And likewise, there are no evidence for extensive serpentinization in the underlying basalts, which could have served as a source of hydrogen needed for the reaction, or for the occurrence of suitable metal catalysts in the dresser tubes. And how does our data reconcile with a biogenic origin? In some of the sample studies, we have also measured the isotopic composition of anchorite carbonate rhombs, and we have found that isotopic fractionation between anchorite, which is the inorganic carbon, and organic matter is in the range of 25 to 33 per mil. And this range is consistent with carbon fixation both by autotrophic organisms using the reductive acetyl-CoA pathways, which are mostly chemoautotrophs, and organisms that utilize the Calvin cycles. So our data cannot distinguish between these two carbon fixation paths, but it is generally consistent with autotrophic microbial carbon fixation. So just a quick conclusion. So we have shown that in situ carbon isotopic analysis in the desiccate reveals submicron heterogeneities which correlate to specific microstructural types. And the range of values is thought to represent the original primary organic matter isotopic heterogeneity. And the correlation between isotopic composition and specific microstructural type is hard to reconcile with solely abiotic origin for the organic matter. And the fractionation between inorganic and organic carbon in the sample is consistent with enzymatic carbon fixation both by photoautotroph and chemolithoautotroph. And I will finish with this artist impression of early life. And I think it's can nicely fit the environment represented by the dresser formation, which could have both photoautotrophs represented by stromatolite and chemoautotrophs, which could have thrived in the hydrothermal environment. Thank you.
All right.
Thank you very much for your presentation. I saw you have schematic view for the formation of the hydrocarbon species on the surface. I just wonder, because you have carbon monoxide and also oxygen on the surface, will that form more stable like carbon dioxide species? Or?
You're talking about the carbon monoxide, which is shown for the Fischer-Tropsch type synthesis. Well, industrial Fischer-Tropsch type synthesis utilize carbon monoxide mostly, but this process was also suggested that could happen with carbon dioxide. So the hydrothermal system, the dresser, probably had a lot of carbon dioxide. I don't know if it had carbon monoxide. This is just as the possibility of abiotic carbon formation by the Fischer-Tropsch type synthesis is just a suggestion. I mean,
okay.
And it was suggested that it can also utilize carbon dioxide. It's just shown it as an example. I don't really think that this process actually happened as I shown.
Okay, thank you.
Any other real quick questions? All right, let's thank the speaker again.